introducing Gyaneshwer Chaubey, a well-known medical geneticist and biological anthropologist. More to that, we take great pride and immense honor in introducing him as the president of Bioclues. I'll just probably quickly introduce him. He obtained his PhD from Estonian Biocenter in 2011, and then he worked for postdoctoral years experience in laboratories. In 2017, he was at the Sanger lab. Then I returned back to India in 2017 to take up this where he started this lab in cytogenetics department of zoology, Banaras Indian University, Varanasi campus. Prior to his PhD, Gyan worked with Laljit in CCMB between 2004 and 2006. And he has had varied interest towards peopling, medical genetics, understanding the admixture population, taking cognizance of various various events that occurred during migration. What is the genetics behind this, and the various biological anthropometrics that his group works on. He has several publications to his credit in Nature, in Science, and in many of those particular wonderful publications. And he has been a member of several prestigious organizations and professional societies, including AIDNet and Lindos Nobel group as well. With this little introduction, I personally thank Gyan Saturday evening. He is usually very, very busy, and despite his busy schedule, he is spending about a couple of hours, for this wonderful session that we're all looking. Thank you much, it's having you. I always learned a lot of things from Gyan. Thank you so much. Yeah, please. Yeah.
Good evening, everyone. Very happy to be here, and thanks to Pras for inviting me for such a wonderful session. The good thing is that we have enough time to talk what we can plan and discuss in this coursework. But what is more important to understand about the Indian population, and that is not other than, not about Africans, not about the Europeans, but mainly on the Indians, that who we are. And if you are working on a medical genetics related with the Indian population, if you are working on the anthropogenic or some anthropological characters which are associated with some positive selection in Indian population, if you are working on negative and keep on switching each other's space because of the advancement in the technology. So these are the basic knowledge which usually are useful to understand the Indian population. We have a number of slides to see and to discuss, and then at the end a genetic tree related with the mitochondrial sequence. So what I would supply you? I would supply you the mitochondrial sequences from different individuals. Then we use a very basic but very useful software that is called as MEGA. And by using the Mega software, we try to see that which sample, which individual is more having and how these individuals in a, all and all associated with one another. So let us start the presentation with a slide. Yes, just wait a minute. I think it should be available soon to all of you. Great. So we have plenty of time, and there is no need to be in hurry. And if you don't understand anything, you can stop me in between, and then we can discuss about those things. So as soon as the new data are coming related with the Indian population, unfortunately we are lagging behind if we compare with the Europeans, Americans.
Sir, voice is not clear. Excuse me, sir. Your voice is not clear.
When I come a bit closer, so do you hear me more?
Some kind of echo kind of thing is there. I think it's not clear.
I can use the headphones. Just give me a minute. I can try with the headphone. Maybe that would be better for you. Just give me 2 minutes. Now, do you hear a bit
Yes,
better?
sir. Comparatively better, sir.
Oh, this is better, right?
Yes, sir. Better.
Oh, okay, perfect, perfect. Okay, so let us start from the question that why many people they talk about Indian population. The screen is already shared. Is that visible to you or not?
Yes, sir, it is visible.
Screen is visible, right?
Yes,
And
sir.
it is
Screen is
also
visible, sir.
moving.
Yes, sir. It's fine, sir.
Perfect, perfect. Yes. So why we are unique? Why we have various kind of normal in other parts of the world, and how these information about the genetic information about these population would help us to understand the disease pattern in India. Take in our presentation and which we take in our discussion, and we try to show you that the Indian population they have very specific traits which are usually not found anywhere in the world, and some are shared with the so here is the social structuring of India. Here you can see that into three major groups. One is the caste group. This is a very unique phenomena in South Asia where we see a hierarchical structuring of the caste. Then there is religious groups. The third group is tribal groups. So all these groups each other, but their subcaste will be same, then their gotra will be same, but still because of the living in two different regions or two different areas, the differences in the genomics would be also quite relevant for that. So the geographically isolated caste and the religious group will also have geographically isolated religious groups. Tribes, they also have geographically isolated tribes. Then there will be subcaste groups. Then there will be regional subcaste based on the geographical structuring. Then there will be Gotra for the caste groups. Then exogamous unit. So the grouping is like here one circle shows one Gotra. So here you can clearly see there is a gene flow between some of the Gotra and most of the Indian population. They follow the patriarchal society. So after the marriages, female goes to the house of male. So even in some of the caste groups, the division of the movement of the female is also not very much fluent. This means that some Gotra they intermarry with very specific Gotras. So the marriages between some Gotras are very specific, and the female gene flow is usually associated with the marriages between two Gotras. Some Gotras they intermarry quite frequently, but some Gotra they don't at all. Like for example, this Gotra and this Gotra they don't intermarry. This means that because of the movement of the females in each and every Gotra, even a caste is endogamous, but still each and every Gotra will have a very unique population structure. Whereas in religious group, the as a caste group. So so these kind of structure, do they have anything special for the genetics? Does it make our genes something special? Yes, they do, because if certain caste group staying in a certain environment, then and eating a very specific food, then this caste group will have the pressure of pathogen living at this region, specific region where this caste group is living. Then the intermarriages practices, which is very, very unique. Then their food habits, which is also very much caste specific or region specific. So all these jointly make our population very unique. And these groups, when they intermarry, then when the total population is huge, then the chances of similar type of genomic segments coming together is usually low. But when population are smaller, then the chances of similar type of segments used to come even by breaking by the recombination. They used to come quite frequently, and that is usually enhanced in in certain cases, and these certain cases are usually caused by the founder effect or some other natural phenomena, which is usually non-natural. When in a case when we reproduce freely, we marry freely, but in this case we don't reproduce freely, we don't marry very freely. So these kind of barriers usually make some of the alleles or some of the mutation frequency higher than the normal. And if this mutation is causing a disease, then usually some of the diseases they run in these specific groups. So what I wanted to tell you that our caste structure is also one of the major factor which keeps profile of our diseases or profile of our genetic structure in a very peculiar way, and this peculiar way makes our caste system very unique, and this caste system makes our genomes very unique. So for example, like in Europe, if someone picks up, let's say, a French guy is randomly being picked up, his genome is being tested. So only one could say that this French guy is belonging to France if there is no recent marriages in his whole pedigree. So, like if someone comes from, let's say, Eastern Europe, from Latvia, Lithuania, or Estonia, and marrying a French guy, then the next few generations, the East European genome would also be mixed with the French genome. So that would make a difficulty for that French guy to identify as a French. But in India, because of our endogamy practices, strict endogamy practices, even each and every caste group has very unique genetic structure. If you look to the mixture graph or to the principal component analysis, you can clearly see that the groups they usually make a very tight cluster, and that is because of the intermarriages which is happening in a very certain limited amount of migration of the females in caste groups. So because of that, this is a picture which shows the plot of homozygous segments which are unusual when the marriages happen freely, when the progeny is very specific for a specific caste, that okay, this caste only marries, this gotra only marries to that, let's say A gotra makes marriage only with the B gotra. So because of that, the population of South Asia usually makes such kind of genetic structure, where you see that the homozygous segments are very much big. They are comparatively much bigger than any other population, and the number of such homozygous segments, which has been plotted on the x-axis, these number of segments, they are usually much higher than we expect. So the expected heterozygosity, expected number of such heterozygous segments are usually much lower in case of populations which are bigger, which don't follow such kind of practices. But in our cases, like some population, for example, like Onges, there are just 104 number of Onge people. So obviously there is a very restricted, and they are also restricted into the Andaman Island. So for them, having a large number of homozygous segments is much more obvious. But a population, for example, like Mudaliyar, Ulladan, Nari Kurwar, Malai Kurwar, these are the South Indian population. For them, they are living at the mainland India. They have not been isolated by any geographical barrier. Even, even though these populations, they carry a very high number of segments, and also their mean length of segments, their length of homozygous segments are much higher than we expect. And here you can see that mostly the South Indian population, they are usually coming out of the bar. Even the Indian Jews population, the Jew population who has migrated some 2000 years back from Middle East to India, they also have a very high homozygous segments because of the cross cousin marriages. The Parsis, the Muslim Jat, and then many other populations, whereas large number of South Asian population, they have low amount of homozygosity because of their very high effective population sizes. So we have all the varieties. And for example, like if some allele which is making a disease for some population living here may not cause the same disease in this population present here. So we have variety, we have variation. Everything is present in India, even the lot of exceptions, which could even break the rules of phylogenetics, break the rules of genomics and genetics. They are also present in South Asia. So the unique population structure due to strict endogamy is one of the major region which divides India into various groups. So in 2005, when we worked with the complete mitochondrial sequence of five Onges, five Great Andamanese, five Nicobaris. So, and we found out that they are the remnants of out of Africa migration, and probably human. They took the southern route and then reached to Andaman Island and further reached up to the Australia. We thought that everything related with the human migration question has been solved. But in 2015, when we have used the complete genome of nearly 430 individuals, then we understood that this story is not so easy. And the modern human origin, the human who have conquered from Africa to rest of the non-African world, and the whole globe, it has covered in a very short time span, which is very phenomenal. And the modern human origin is not so easy to understand, and there are a lot many things which are still being discovered. So at this moment, like if we feel that the human and chimp, they had a common ancestor something like 6.5 million year ago, then the human branch, it has given another branch around 800,000 years back. And this common branch has again separated into two branches, that is 640,000 years ago. And then it has given rise to hominin, that is the extinct humans, Neanderthals and Denisovans. And the line which has given rise the modern human, has probably made a modern human something like 200,000 years back. And this 200,000 years back, modern human lived in Africa. Then there is not just single out of Africa migration, there had been several, but at this moment we could track up to two out of Africa migration. One has happened something like 90 to 120, and then the major migration has happened 40 to 60,000 years back, which has given rise the all non-African modern humans. So when there was information of the genomes available up to the level of mitochondrial DNA, up to the level of few SNPs of Y chromosome, then it was very easy to reconstruct the population tree, and it was 100% said by most of the population geneticists that the modern human originated in Africa, then went to the rest of the part of the world by replacing most of the archaic humans. So the replacement theory was the most predominant theory that has been accepted by the people. But as soon as new data sets started to come, and as we dig deeper, which is a very common phenomenon, as we dig deeper, things are much more complex than we have ever thought. So human origin is not so easy. There are a lot of migration, there are a lot of mixing, and not only the modern human has originated in Africa, but it also mixed with Neanderthals and Denisovans, a common ancestor which got separated around 804,000 years back, again mixed with modern humans something like 40,000 years back, and they exchanged their genes, and now we have around 1 to 4%, 1 to 3% of the Neanderthal gene, and some population even they have higher, and also like Melanesians and East Asians, they have Denisovan genes. So these archaic humans, they are only known, but still there might have been several ghost population which are yet to be discovered, and we are not just, we don't have just a single kind of DNA structure. We have a mosaic of DNA structure which is mix of not only modern human, that is the major ancestry, but also the Neanderthals, Denisovans, and many more ghost population. So our origin is much more complex than we have ever thought. So what are the basic criteria? What are the basic structures which usually we use to define the human population? But before that, I think we should go to this slide. So a normal human body is made up of around 37.2 trillions of the cell, and each and every cell has one nucleus, then cytoplasm. There are many cell organelles, but mitochondria, here they have been shown. Why? Because mitochondria, there is a mitochondrial DNA, and mitochondria is exclusively passed from mother to son or mother to daughter, whereas in nucleus we have 22 pairs of chromosomes. Those are autosomes. Then there is 23rd one is sex chromosome. X comes from female. X also comes from the male to the son and to the daughter as well. But Y chromosome exclusively comes from the father. So if one has to study the mitochondrial DNA, the Y chromosome, and the autosomes, then let us see that how they help us to understand our history. So this is a pedigree. Here you can see that this is a girl. There is a mother, there is father. Then mother side is grandmother that we call as Nani. Grandfather that we call as Nana. Here we call Dada. Here we call Dadi. And this is how we can go back to the several generations. But if we study only mitochondrial DNA, then we could get information about the green color people. Like if you study mitochondrial DNA, then mother, grandmother, nani, great grandmother, great great grandmother. So this is how we can go back up to the time of Eve. And And also, one very interesting thing is that the mitochondria pass from one generation to next generation. That is 100% of mitochondria pass from one generation to another. There is no recombination in mitochondrial DNA. So, at the male line, if we study the Y chromosome, we could get information about the blue color people, the person's father, grandfather, great grandfather, great great grandfather. So this also is one of the phenomena very common in South Asia. When we study the Y chromosome, then usually we track the gotra of a individual. So, but now just by studying the mitochondrial DNA, just by studying the Y chromosome, we could get information about these people. Only the blue colored, only the green colored. What about these gray colored people? Because in our pedigree, we have several ancestors, not just a single ancestor. We have several ancestors back in the pedigree. So the third thing is autosomes. Autosomes comes from both of our parents. 50% usually is being donated by the father, 50% is being donated by the mother. So, what happens when we go back to several generations? As we move step by step, each and every generation back, usually we lose 50% of the genomic similarity, the genomic, not similarity, but exactly the same genomic segments. So, and this is known as identity by descent, IBD. So, by studying biparental markers, that is, the markers which are present at the chromosome 1 to 22, the autosomes, which half we receive from father, half we receive from mother, we could get information about all the ancestors. But the problem is, as we go back, we lose 50% of the information. So that is why for a population genetic study, usually people use details about all three kind of markers. The mitochondrial DNA, which can trace straight way to the maternal site, Y chromosome, which can straightway go back to the gotra or paternal site, and autosomes, which can go back to several generations, but which can get information, which can give information about the all of your ancestors present in your tree in the past. So the ancestors who have contributed to any segment of your DNA, if you study the autosomes, it is highly likely that you can find out that ancestry. And one very good example is Neanderthal ancestry. When people have studied the mitochondrial DNA of modern human, Y chromosome of modern human, there was not at all, there was no sign, no trace of Neanderthal related ancestry. But as soon as they studied the autosomes, the biparental markers, millions of markers for the 1 to 22 chromosomes, they found out that a very significant amount of Neanderthal ancestry was present in modern human. And even there are a lot of studies, there are a few more studies which are coming up, very exciting studies, which are telling us that we got even certain traits from the Neanderthal which are quite useful for us to survive in such a vivid environment which is present into the rest of the non-African regions. So So the humans, the modern human ancestors were very smart. They have received the good things from the archaic humans, but no one knows that how these archaic humans they have completely disappeared from the globe. So initially we discussed about the replacement theory, but now this replacement theory has been revised, and now they say this as a leaky replacement theory. So, so the leaky replacement theories are the basic concept now which are being used. So let us come back to the two basic tools which we usually use to understand the population structure. Either it's a disease study, either it's a population genetics study, or any kind of study, any marker, if you are studying, then these are two basic tools people they use for sanity check, to understand the population structure, to see the genetic components which are present in the population. So the first method is principal component analysis. And if you would have done any genome-wide association studies, if you would have done any population genetic study, you must be familiar with the principal component analysis. The principal component analysis is a method which is usually the problem of humans, that human cannot see multi dimensions. So we need to break our data. We need to not break, even join our data in such a manner so that by losing minimum information, we could show maximum. So human is very good for the two dimension. So usually the principal component analysis is breaking first the population into multiple dimensions, then joining it into two dimension to show the population structure. So that is also a very interesting part of the principal component analysis. What it uses the allele frequency data, the alleles which have been found into multiple individuals. It calculates the frequency of those allele in the population, and then look for the similarity, dissimilarity, then the pairwise differences, and this is how the principal component analysis is being made. But there is another question that when you see such kind of a allele or distribution of a certain traits, then you cannot differentiate this with the isolation by distance model or with the migration model. And there is also another term called allele surfing. Like when a allele moves from one population to another population without the migration of population, then this allele is actually associated, can be explained by the isolation by distance model. So this isolation by distance model says that we share more allele with our neighbors comparing to any distant neighbor. But this is not at all, I mean, not 100% applies for the Indian population. For example, like Indian villages, where you have several caste groups, because these caste groups, they don't interchange the genes, then a certain caste will be more closer to the same caste in different village comparing to a different caste of the same village. So, but in a longer term, overall, the the Indian population history or Indian population genomics is usually defined by the geography. For example, like someone is who is living in Punjab and someone is living in Tamil Nadu, they are not different that because they are speaking different languages. One is Indo-Aryan, another is Dravidian language, but because they are living far apart from each other and then marrying in their local community. So this helped to to increase the private alleles in these population comparing to the shared allele which has been founded by the founding population, and these founding population usually comes from a single group or even not more than 3 to 4 groups. And then second analysis is associated with the admixture analysis. Admixture analysis is actually the supporting analysis with the principal component analysis. It uses the maximum likelihood method to estimate the individual ancestry or population wise ancestry with the colors. And these color codes are usually defined by a certain kind of ancestry, and this color code, when they are present higher in the same population, lower in another population which are distant to them. So based on that, usually the algorithm and then the optimization of these specific algorithms usually help us to understand the population structure and the presence of a certain ancestry component in a group of individuals. So, sorry. So this is actually based on the very classic program that is a structure that has been developed by Jonathan Pritchard during year 2000, when people used to use the forensic markers, the STR markers. Uh, so based on these STR markers, they have designed the structure program. But the structure program, because it could not handle the multi-locus millions of SNPs, then they have designed another program that is admixture, which could handle even millions of the SNPs and could run the program in a very short time. So we have already seen this. Yeah, now this one. So this is a very complex process to decide that what kind of analysis is actually needed to understand a certain population history or to understand a disease pattern. But some of the analysis are very fundamental, very basic. For example, PC and admixture. Usually, they have been done in each and every kind of question to just to understand the basic information, and this basic information usually comes from these two programs. So, also the understanding the mutations, and by using these mutations, one could also reconstruct the tree. So these kind of the tree usually comes, they are the outcome of the mutation. So these mutation usually decide that whether a tree is a smaller, whether a tree is a bigger. When it is a smaller, then what it could be. When it is bigger, then what it could be. So for example, like a certain segment of the DNA which is present in several individuals is known as haplotype. And if there is a change in any nucleotide position of this haplotype, that is called mutation. This mutation is also known as single nucleotide variant, SNV. But when it is more than 1%, the frequency of this mutation is more than 1%, then it will be known as single nucleotide polymorphism. So the single nucleotide polymorphism is nothing. It is just the polymorphic variance. Variance means mutation or single. If it is just a single nucleotide change, then it is better to call it single nucleotide variant than a mutation. And when these haplotype, they are present in a huge number of individuals, large number of individuals, similar kind of mutation, then they are known as haplogroup. For example, like haplogroup. We have just given it as a name, that is haplogroup YY, because it has a mutation at this position and this position, and XX when it has mutation at this position and this position. So now we have two trees. One is bigger, another is smaller. Bigger because it took more time to accumulate the mutation. So when mutation has been accumulated, then the tree will be bigger, the branches will be bigger. And when mutation will be less, mutation will be less number of mutations will be present, then tree will be smaller. So more mutation, more diversity, the large sum of branch lengths, and more time for mutation to accumulate. Then it has less diversity, small sum of branches. Even if you use the scale and start measuring these branches, you would see that these branches are smaller comparing to these. So it might have taken less time for the mutation to accumulate. But then there is another phenomena, that is genetic drift. For example, like even the Andaman Islander population is living there since at least 25 to 30,000, even more. You would see a smaller branch comparing to some mainland population living in India. But possibly this is because of the drift, because drift keeps on working on a smaller population and it tries to randomly reduce the frequency of allele. But when the population is less, then this happens more frequently, and because of this is a very different phenomena, but I don't think this is needed to explain here. So now by using these information, how and what we can infer about the Indian population. So I give you some of the key studies which have used this kind of information, and by using these information, these studies have helped us to understand more and more about the Indian population. So this was one of the very interesting study related with the Himalayan population. So Himalayan population, and one very important thing is sample collection. So there are two ways, either to understand about the population, either you collect each and everyone present in the population, but that will be too expensive to genotype everyone. Then what is the second thing? Second thing is the population phenomena says that if we go random collection, collect a population living in a certain area, randomly collect people, avoid the three generation, then by collecting around 50 to 100 individuals would be enough to show the whole ancestry of the population. And even with the latest technology, just a single individual, I'm talking about India, single individual of a particular caste group or tribal group or religious group with the complete sequencing, complete genomic sequencing, you can get the most of the information about that particular population from which this guy is belonging or the girl is belonging. So the technological advancement is much more helpful to understand the population history. So this was Himalayan and the adjoining population, people who are living at the Himalayan region. This is a very unique area, and it has also acted uh, as a refugium for the early human migration. Because early humans came, then there was a last glacial maxima to the Europe and to the Middle East. Then it was difficult for the humans to live there. Then many of them they lived into the Himalaya, and then and when the climate was favorable, then human again came out of that and then expanded further. So the Himalayan and adjoining population were sampled. This is the example of principal component analysis, and here you need to understand this. This is principal component 1 that explain around 6% of the ancestry. Principal component 2 around 2% of the ancestry. So
Sir,
here are the
PCS slide is not visible. Still, we are in sample the population.
you cannot hear again.
No, sir, no, sir. Actually, slide. I am talking about the slide. Still, we are
So
in the
slides
sample the population. We are not seeing PCS slide.
Oh, and now
Not yet, sir.
what can I do? Can I restart it or what? Because it is moving in my computer.
Actually, there is a lag, sir.
No. Slide is moving. It is seen.
Slide is moving, right?
Yeah. Now you are in sampled population, no, with the Himalayan and.
That's what I am also saying. But sir is explaining PCA now. Sampled population. Still, we are seeing sampled populations, but sir is explaining PCA. PCA slide.
So can you see this principal component analysis slide or not?
No, sir. No, sir. We are still in sampled populations.
Okay, then I can restart it. Let's wait just a minute.
Yeah, Gyan, if you
Let's
could restart. Yasin, I think there is
Yes, sir.
a small lag actually.
Yes, sir, yes, sir.
As Mustaq says, it's moving, but there's a small lag.
Yes, sir. Yes, sir.
Yeah, because of the internet problem, I stayed in the lab so that it should not happen. But I'm sorry that it is still happening here. Okay, let me try again. Let's look it. No problem. We have plenty of time, so don't worry. The important thing is we need
Sir,
to understand
one thing we can do is we can all switch off videos. Then.
Oh yeah. That I can also do. Yes.
Okay.
Now, do you see the
Yes,
principal
sir. Now we
component
are
analysis?
seeing PCA. Yes.
Perfect. Okay. So this is the principal component analysis that explain the placement of Eurasian population over the principal component 1 and principal component 2. So here are the European population, the Caucasus, the Middle East, then here are the Pakistani, here are the Indian population, Indo-European, Dravidian, they are mixed here. So by seeing this line arc line, you can correlate very well with the geography. The European population, the Caucasus population, the Middle Eastern population, they are just above at the left side of India. Then there is Pakistan, then the North Indian population, then here is South Indian population. Then these are the Indian Austroasiatic speakers, which are going out of the line because of their Southeast and East Asian related ancestry. So what I wanted to show here that the individuals which are present here are the individuals which have been placed here. They usually carry their geographical placement. Some of the population, based on their ancestries, they usually also show the mixture of ancestry, that is because of the recent gene flow or because of their migration. For example, like Himalayan and adjoining population, they go closer to the East and Southeast Asian population rather than Indian, mainland Indian population. And here one can see that there is one client that represent the East and Southeast Asian population, but there is another client. These are the Himalayan population. They are in between East Asia and India, so they are between China and India population, but they are more closer to Chinese group. So, and this is the another basic analysis which I showed in the beginning, showing that there are two basic analyses. One is principal component, another is admixture analysis. So in admixture analysis, we see the different colors, and these colors actually show the ancestry components, and these colors are associated with a population group, with a regional group, with a particular geography. For example, like the African population, they have their own color. Then there is a Middle Eastern population. Then there is these are the African population. There are two colors in African population up to the Yorubans. Then there are these are the Middle Eastern population up to, and the West Asian population. They have up to three colors. And then there are European population. They have their own predominant color, but there are as well as some more colors presenting to them. Then there are the Caucasus population. Then there are Central Asian population similarly. Then there are South Asian, including Pakistan and Indian population. So here, what is important to show here, like the Indian population, they have two major colors. One is dark green, and another is light green. You can see that this dark green is very exclusive to South Asia, and by David Reich and by Dr. Thangaraj and Lalji Singh group, they call this group as ancestral South Indian population. And then there is a light green population that is known as ancestral North Indian population, but that is not restricted to only North India. That is also present into Central Asia, the Caucasus, also into the Middle Eastern population and some European population. But about the Himalayan population, you can see a very unique color, and that is present into Himalayan, very predominantly present into the Himalayan and adjoining population. And this is also present in some of the part of East Asia, and up to the Siberia, this ancestry was present. And what is interesting to see here, show here, that the Himalayan ancestry, that is red color, means quite high amount of ancestry is here. That is more dispersed towards the east, not towards the west. If you look to the mainland Indian population, this ancestry was not coming towards down to the Himalaya, but this ancestry was going other side of the Himalaya and up to the Siberia. This is ancestry is quite predominant. So that is why I told earlier that these area has actually acted as a refugium for many human population, and because this region was quite cold during the last glacial maxima, so as soon as this became a bit warmer, the people have moved from this direction to this direction. And these are other statistical tests which shows that how is the direction of the gene flow, in which direction. So these kind of analysis which have been done. And this is another interesting part I wanted to show you. This is associated with the haplotype based analysis. So there are two kind of analysis. One is allele frequency based analysis. Second is haplotype based analysis. So in haplotype based analysis, and why people say that haplotype based analysis are much better than allele frequency based analysis is that Prash, when do we need to finish? 8 o'clock or 7:30?
No, you can take your time, Gyan. We usually start our hands-on at 7:45.
Yeah, because
So it's up to
30
you,
minutes
please.
we would take for the hands-on training. Okay, can you please repeat? I could not hear correctly.
At 7:45. Yeah, yeah.
7:45. So 15 minutes
What I meant
we
was
still have.
30 minutes, Gyan. You can start at 7:45 or 8 o'clock. That's up to you, please.
Okay. Okay, perfect. Perfect. Perfect.
Yeah. Please. Yeah. Please. Yeah. Thank you.
Okay, so this is one of the analysis where you could see that how haplotypes has been shared between two populations. So usually on the left side, these populations are the donors. So we wanted to see that how Himalayan population they have received a haplotype, a very core haplotype from these populations. So red color means more haplotype, the gray color means less haplotype. And here you could see again that with the Southeast Asia and East Asia, they have donated a huge amount of haplotypes to them. And here you can see that the population sharing, the red color means the population like Sherpa. They are the very unique population of the Himalayan region with very high frequency of the high altitude EPAS1 gene adaptation. So these population, because they are smaller, they are very highly mixing with within the group. So this is why they have a very unique pattern, which is presented to these population. So, and what was another interesting point that with the Siberian region, the Himalayan population share more than with the Southeast Asian population. So this is against the geographical clustering because usually the Himalayan population should share more with the Burmese or Cambodian than with the Koryak, Nagasan, Buryat. These are the Siberian population, but this is other way around. They share more with the Siberian population. It means there is a close genetic connection, but that is very old with the Himalayan population and with the Far East means Siberian population. So these kind of analysis usually help us to understand. And this is again, you can see that runs of homozygosity, the huge amount of segments, and the number of segments are quite high for the Sherpa and the Kusundas, Eonagas, and Khasi populations. And there is also a way to calculate the archaic ancestry, the Denisovan ancestry, and you can clearly see that some of the Himalayan population they have a very high amount of Denisovan ancestry comparing to the other northeastern population. So this is another connection between the Denisovan and the Himalayan population. Then initially I discussed about the population of positive selection, that there are some traits which are usually being selected by the nature, and in no time, within a very limited time, the frequency of these allele usually become 100% in the population. So for example, there is a very interesting gene, that is EDAR gene, and nature is also very unique because and that we also see in our Indian scenario. And then when either in university or in overall life scenario, when someone used to take care for a job in a nicer way, in a much better way, then usually we also give another job to the same person because we know that that person would usually do that job in a proper way. So nature also follows the same criteria. EDAR gene was selected for some trait, then nature started to give it many more traits, and at this moment there are at least 7 or 8 traits which have been discovered for the EDAR gene. So this is known as pleiotropic gene. So one gene which can control several characters. So here you can see that it has a very high frequency into this region, but in India, only few population who have associated ancestry with them. Only those population they have this kind of like Tharus. They also have a limited East Asian and other side of Himalayan ancestry. So that's why there is a bit higher frequency of this allele. Otherwise, other Indian population like the Dravidian population other Indo-European population, they have nearly zero frequency of this allele. So the positive selection was predominant into East and Southeast Asia, but it did not work out in South Asia. This is another enigma, and people are trying to solve this question that some trait was positively selected in one region, but in another region there was nothing happened to this trait, and because of that, only few limited population has been found to have this kind of trait. So this is another analysis which shows that when the population admixture has happened, so this is also possible with the current available technology and available data sets. And there was another phenomena with the Y chromosomal markers that when we go up, frequency of certain haplogroup increases, and when we go down, frequency of other haplogroup increases. So these two haplogroups played a very antagonistic role for each other. When this increases, this goes down. When this goes up, this goes down. So this is a very unique kind of very special phenomena which we have observed in the Himalayan population. This is another story related with the Romas, the gypsy population who migrated out of India to Europe, and that has happened something like 1,500 years back. And because of the population coverage, because the CCMB collection was nearly 25,000 samples, other people, other papers, usually done by European authors, they could just pinpoint a region. But because of the very good population coverage, we could even pinpoint the northwest India, and mostly the scheduled caste and scheduled tribe population, who are actually, they share the very close common ancestry with the Roma population. And that even the dating shows that there was a haplogroup H which has originated somewhere in the south, dispersed all over the India, but just 1,400 years back, a group has migrated out of India carrying the signature of this haplogroup. So, and then there are two Dharkars and Kanjars. These are two gypsy Indian population. They share a very close common haplotype with the Roma population. So that shows that what is the power of the markers, power of the assembly of the genes, and then the power of selection of the markers. Usually these things help to pinpoint the origin of a population. Like the Jews population, there were two migration of the 2,000 years back. Mumbai Jews came around 1,100 years back, and these two groups clearly can be traced back with the genomic analysis. This blue color ancestry connects these population. For example, here, this blue color ancestry is actually Jewish ancestry, and that is present only among Mumbai Jews and Cochin Jews. But the amount is quite low, so it means that a very limited number of people came. They assimilated some of the local people, and now they are following the strict endogamy practices. So these kind of analysis have been done with the Indian population. The Siddhi population analysis has been also done, where Dr. Mustaq was also involved. And there you can clearly see that this is Africa, this is India. And you can even use the scale to measure the percentage of ancestry. Like you can clearly see that this one is closer to this, and this one is a bit further away from this. So the ancestry is just opposite to the scaling distances. I mean, this is not 100% correct, but as actually one can measure this way. For example, this is more closer to this. It means 65% of the ancestry of Siddhi comes from Africa, and around uh, 35 to 40% of the ancestry comes from from the South Asia. So this is how important is the PCA analysis, which could even clearly show this kind of connection. The Parsis, who migrated 1200 years back, similarly, these ancestries, but there are some caveats here. For example, like the Parsis are here in between Iranian population and in between the Pakistani population. This does not mean that they have been derived by Iranian and Pakistani population, but because they have ancestry associated with South Asia, ancestry associated with Iran, so that's why they fall here. So one has to understand these kind of caveats and this kind of explanation. So here are Iranian, here are Parsis, and this blue color is exclusively present in the Parsis, but not into their neighboring Indian population. And the dating of the genomic dating clearly showed the same time, which usually being defined by the Parsi migration. And this is another runs of homozygosity and the uniparental analysis, which actually showed a very clear-cut relation of the Parsi population with India. So these are the basic question usually which we have taken, and then we have discussed with the South Asian population and South Asian ancestry. So each and every population, you can see that the population, they make the colors and the clusters, they are very nicely clustered together. They are very nicely made up in the same way. I wanted to show you this slide just to understand and thus to kill many of the historic notions. For example, like these are the North Indian and these are the South Indians, and these colors show their ancestry components. So this blue color is the Indus Valley component, and then the green color is associated with the Andaman Islanders, and then this dark blue color is associated with a hunter-gatherer population which was present into the South Indian region. So here you can see the founding population of either South Indian population or North Indian population. They are the same, except the proportion of various founding population are different in these two populations. So there were many questions, and one of the very important question was the related with the mystery of Ajnala people. So the Ajnala people, there were some skeleton which have been discovered in 2014, just based on some historical knowledge or some of the abundant book, and this was discovered by one of the local historian who lived in Ajnala. He discovered it, but then after the discovery, there had been several theories. Many say they are the Pathan people, they are the local, they are the British, because the bone doesn't have any skin color. So many say that they are British killed by Indian rebellions. Many say that they are victims of 1947 Indo-Pak separation. There were a lot of speculation about these groups. We have used the genetic analysis, we have used their mitochondrial analysis to get the oxygen carbon isotope analysis to understand their food habit. See, their teeth was such a beautiful, uh, beautifully preserved. So overall, if we look to the ancestry of South Asian population, one is very unique to South Asia. Another is associated with the West Eurasian population. And third one is associated with the East Eurasian population. And here, if we plot the Ajnala population and look for the closest match, then they show a very close match with the Ganga plain population comparing to Punjabis, Pakistani, Central Indian, and South Indian population. And the principal component analysis placed them with the Bihar, Jharkhand, Uttar Pradesh, and West Bengal population. So this has confirmed, and also the food habit was also from the Ganga plain region. So this has confirmed that this population has been, they were the revolt. They have done actually revolt with the British regime during 1857 time. They have been killed mercilessly by the British people, and finally their DNA has helped us to bring this story, which was completely abandoned by the people. So these kind of work usually cannot be done just with a smaller group, a smaller sample size. So we collaborate in a larger area, larger number of people all over the world, and thanks to them to make this kind of study possible. So thank you very much. And if we have some question, then we can discuss, and then after 5-10 minutes, we can go for the
Sir,
our
it's an
exercise.
amazing lecture. I remember that in 2005 you have written something in science, and I
Uh-huh.
was a PhD student. I read your paper and I was so interested. Oh, our Indian people are also writing in science, nature kind of things. So I'm just big fan of you. I was not knowing that you are in our group. Uh, so I'm happy to meet you in this training, because earlier also we had that kind of interaction, but now only after seeing your presentation only I could recollect that that is you. So I'm still excited because I was having that excitement after seeing the paper when I was a PhD student. I'm feeling the same thing now. I'm so happy, sir.
No, thanks for your kind appreciation, but like the common, we are also very common Indian people. I think each and everyone have some unique personality. Once upon a time, I lost all my interest for the further studies, and then I did not have any way to go further. But then just with a reading by a single research paper helped me to move in this direction, and I'm happy that I could do and I could contribute something for the nation.
Sir, actually, I was preparing for civil services,
Uh-huh.
but after seeing some of papers like yours, means not only your paper, but some other papers also. So only
Yeah,
those
yeah,
things
yeah.
motivated me to be in science. I got selected for DSP post. I left that and I joined in ARS.
Oh,
So
wonderful,
you are
wonderful.
also one among the reason for that.
Oh my God, that's amazing. I think this is one of the science is most underrated job. People
Yes, sir.
they know that we are scientists, but what we do, how we do, they don't have such kind of interest. I mean, being a celebrity in science is completely impossible, except for some like Lalji Singh and then Sri Abdul Kalam ji. Otherwise, publishing many papers, doing something for the nation is usually one thing which I think everyone should do and should think in this direction. And it does not matter that whether a scientist is a celebrity or scientist becomes a sensation at the YouTube channel or sensation at the social media. But there are many scientists who have worked for India, who have worked for the tirelessly for 24 hours, and then they 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 got nothing, but still they were happy because they contributed something. So I think
So they
we
are
should, yeah,
recognized, they are followed by students like, okay, now I am following you. Just like that, there are so many people will be following me or some other scientists who are sincerely
ex-
working. So that is the
exactly,
recognition. That
exactly,
is the recognition.
exactly.
So they are not known to the common people just like politicians, but they are known to the scientific group. So anyway, you are a celebrity, sir. We will accept it.
No, no, that's okay. Yeah. Do we have some question, and then we move further for the making the phylogenetic tree?
Yes, sir. We will move. I don't have any question. If somebody else has.
Sir,
Yes.
actually, when we are talking about, sir, myself, Dr. Vipul, I'm from Forest Biotechnology.
Yes,
Sir,
actually.
in many legal case, people are using microsatellites to
Mhm.
identification of the victims in case of rape or some murder. So at that
Yeah.
time, whatever microsatellite loci they are considering, is it how they are deciding? Because means for Indian population and for some Eurasian population and for African population. So how they end up in the conclusion? So that,
Yeah, yeah. So, these most of the kits which have been designed by the commercial companies, they have been developed on the Europeans. So, I give you a very, naive kind of example. Like the European population, if you compare with the India, they are less diverse. So Africa is most diverse, then India is second, then other regions, and European is less diverse because of their climate and their late and lot of replacement of the population in the population history. So when something has been discovered on a population which is less diverse, then it would automatically work on a population which is highly diverse. So the differentiation of the population, let's say, that's why they use not just one single loci, single locus, but they use multiple loci. So these multiple loci, if one loci is monomorphic, then it is not necessary that other loci will be monomorphic. It may be polymorphic. So in such cases, usually most of the loci which have been discovered so far, they are polymorphic. But yes, there is a need to make Indian specific microsatellite markers, which has not yet been done. And the most problematic thing for forensic is to develop the databases. Why forensic is not able to use the latest technology? Because of the lack of the databases. So database for microsatellite at the moment is in a much better stage. So that's why people use the microsatellites. There are many new techniques. Only one has to do it randomly for a large number of population. Then that techniques could be even much better for the population identification or the individual identification. So population genetics and forensic genetics, there is just a single difference that population genetics needs to find the common variant and forensic genetics needs to find a unique variant that is individually specific.
okay, sir. Means actually, I came up with that question because in case of timber tracking, you must be knowing, sir. So the forest-specific timber identification in case of some legal matters of like red center and related. So they have to develop the panel for that forest, and then after it is possible to employ for tracking through forensic means. So I was curious
Right.
for this human since after
Yeah, this should be done for actually each and every part of the science, but I think we need more and more people to work in this direction.
thank you very much.
Sir, I do have one answer for the timber related one. Like we
Yes.
are doing fingerprinting from our institute, fingerprinting for varieties. So the same kind of thing, the forestry institute is, forest council is also doing the same kind of thing. So they do have something to identify the legal issues. So they can identify the wood samples and then they will be able to tell what kind of tree or from where it is collected. So because the region wise also they can tell. So that much information is available, at least for very important species, something like sandalwood or red sandalwood, those kind of trees we do have data.
Correct. Yeah. I agree, and I think these areas also need to be developed in a proper manner.
Sir, one more question. I don't want to miss the chance since I'm with you, sir, right now. So, sir, since DNA extraction is very problematic in some matrices in case of plant, but as far as
Mhm.
since you are working with this fossil or some, you show some some very ancient type of bones and everything. So means how efficient this method is to extract the DNA, or are they using some special kind of chemical or any technology by which they can?
Yes, yes. This this ancient DNA technology has developed quite a lot now. As you can assume that a sample, just a pinky finger bone, has been discovered somewhere in Denison cave, and just by sequencing this pinky bone, they have discovered the whole human line, that is a Denison line. So the powerfulness of this technique has grown enormously. But again, the problem is with India, that Indian climate is not favorable to preserve the ancient DNA. The soil is very acidic, which is not good to preserve DNA. But still, there are development. There have been things they are going on in this direction, and hopefully within few years has their own optimization. And also in the sequencing technology, they usually consider only transversons. Not the postmortem damage or any such kind of damages. So they may get the false signals. So usually the transversons are true signals, and usually they compare those mutations. So they do lose some of the lot of positions, but still they get the correct answers.
Okay, thank you very much.
Okay, so we go for our exercise. I want you all to do this exercise because this is a fun. I'm sharing my screen first. So my screen is visible to you.
Yes, sir, visible. There is mega folder.
Perfect. Perfect. Yeah. So if you don't have this software in your computer, then you can just click it and then install this software. If you do have already this software installed in your computer, you don't need to do anything. Just open the program. So I open the program Mega. And there is a tutorial, Mega Part 1, Mega Part 2. I would
Sir,
suggest you,
sir, we
or the
need
link is up. You just go up, go up, go up. There is a link which I have posted at 6:26 p.m. drive.google.com.
sir, please share it again because
I can,
we
okay,
need.
okay, okay, no problem. Just give me a minute. I will share the link here. Oh, yes, it has been already shared. Thanks, Durga. So when you click on this, you will see the same folder. There are Mega Part 1, Mega Part 2. Mega Part 1, Mega Part 2 is for your use only. You can use this tutorial to understand that what you need to do. And the fourth number is the software installation that you can install in your computer. There is a file called mega.crs. I open this file. So this file is a Cambridge reference sequence. That is a mitochondrial reference sequence, 16,569 base pair sequence that you can see here, that you need to use. And then there is a file called sequence.fast. That is a FASTA file. I open this file too to show you the so these are individuals like 289, and this is the sequence 290. This is the sequence. So we have few individuals is to make a phylogenetic tree of these individuals. So by for making the phylogenetic tree, we use the mega program. Because and if you don't have, you can install it. This is not a malware or any kind of thing. You can trust on this link and then install that software. So the full name and that you can also download online. If you have installed the software, just click on Align. To choose create a new alignment. Create a new alignment. Including the, I show you here, better to go back.
Excuse me, sir, it's asking DNA or protein, which one to choose from that?
It's DNA. So I hope you can see the Mega software at my screen.
Yes, sir.
Go, go to align. Number one, edit or build alignment. Then wait for that. Create a new alignment. Click OK. Or go to edit, insert sequence from file. Go to the folder where you have kept the downloaded.
So file name is sick.
So did you
Yes, sir.
download the
File
files?
name is sick.
So SIG, yeah, just take the SIG, click open. Then you may see a window like that. Now go to alignment. First select all. So you need to control A, select all. Okay. This will make the alignment. So after the alignment, you need to go to the data, export alignment, mega format. Click on that. It may ask input title of the data. You can write anything, whatever you want. Protein coding nucleotide sequence data. Just
Can you show the steps after the cluster W? Because I'm just following on this software, so I just missed what you did. Excuse me, sir. You are muted, I think. Your voice is not coming, sir. So you are on mute, sir. Please unmute.
Sorry, this is. Yeah. Is that okay now?
Yes, sir.
Okay, perfect. So align by cluster W, select all. No need to change anything in these parameters. Just click OK. It will align the sequences. And then go to the data, export alignment, mega format. Alignment. Save. It may ask input title of the data. You can write the same alignment here. Click OK. Protein coding. None. Everyone has done up to this level?
Yes, sir.
Yes, sir.
Is there any problem to reach this level?
No, sir.
Yes, sir.
Okay, so now double click on this alignment file, whatever name you have saved. Then this file will open in Mega. You may see something like this, TA, and then close data. And you need to click on phylogenetic tree. You may choose whatever you want, maximum likelihood tree or never joining tree or minimum evolution tree or UPGMA tree. I have chosen the never joining tree. There will be a smaller window. You just need to click OK. अच्छा ठीक. So I'm again repeating one more time. Go to the align, create a new alignment, click OK. Are you building a DNA or protein sequence? Click DNA. Now there is a sequence 1. This is a blank sequence. You don't need to keep this. Just delete this. No need to keep this blank sequence. Insert sequence from file. The sequence which has supplied a CQ. Click open. Control A, alignment, W, click OK. Go to the data, export alignment, mega format, give it a name, some name, and then save in the same folder. No protein coding sequence. You may close after that, close everything, and can go to the same folder where you have saved your alignment file. Just double click there. This will open your file. Click on the tree, phylogenetic tree. I'm clicking on neighbor joining. Clicking OK. After clicking, it should show a tree. I think that is not visible in this version, I guess. I would make us.
Sir, we got the tree.
Yes, sir.
You
I
got the
saw
tree?
Yes, sir.
Yes, sir.
Perfect. Very simple method. Then so let me open the tree. Then I can explain that how to see the relation of these trees, relation of the each and every sample.
Can we share our image, sir?
Sorry?
Can we share the screen to show you the image what we
Yeah,
have?
that would be great. Maybe I can explain in that screen. In the meantime, neighbor joining is compute. I'm using bootstrap, sorry. So there are multiple ways to make these trees by using the various options here. We can use the circular tree like that. The branch style would be something like that. Then click on these. Then you can even make curved style here. Okay, now let me see the.
Sir, in my case, it's showing distance calculation failed.
So I stop the presentation and now you can share the screen. Then I tell you the problems, what you need to do. If someone has problem, then I can tell you the way to rectify it.
Okay, sir.
Sir, this is the image we got.
Yes, perfect. So this is one way to show the tree. You can go to the view.
Sir, we just wanted to understand what it is actually. Exactly these numbers.
Yeah, so these are the individuals. Okay. So you are comparing like 289, 290, 304, 326, 300. These all are individuals, and you are comparing these individuals based on their mutations. Okay. These are the mitochondrial DNA hypervariable segment 1 data. Clear?
Okay.
So,
Sir, if this is 0 or 1627, what this means, and how to infer from the distance, like
I
how
think you have used the bootstrap method. So
yes,
this is
sir.
why you see the 0, 1, 22, 43, 40. at
Yes, sir.
the junction. So
Yes, sir.
these are I don't know how many bootstrap you have used, but looks like you
500.
have used 500. Okay. So this means that you planted 500 types of trees. Okay.
Yes, sir.
And out of 500 types of tree, how many times this 310 and 319 comes together? So 76 times this 310 and 319 comes together.
Okay.
Here 43 times 329 and 334 comes together. So when more bootstrap value means your tree is more robust.
Robust.
Less bootstrap means value means the tree is not very robust. For example, like here you have 0.
Yes,
0
sir.
means that 296 may have a closer relation with 321 than 329, but this is not clear with this tree. Okay? But the like 40 number is more than 0, so it means that 297 and 335 may have very close genetic relations.
Okay.
And here you see that some of the individuals they have big branch, some of the individual have a smaller branch. So big branch means they have accumulated more private mutations.
Okay.
And a smaller branch means they have accumulated less private mutations. So 3110 and 319 are coming just here in this branch. It means they are most closest to each other and they share a very large number of common mutations. And if someone asks you like how closer is 312 with 309 comparing to 327? So you can measure here the distance. 312 is more closer to 309 comparing to 327 because that is far apart from 309. Got it?
Means this is closer to this one.
Yeah, so you can use the distance branch length. For example, like 305 and 309. So they, because they share a same branch, so this one goes here, this one goes here. So it means they both are closer to each other. And then these branches, they join together and then they share another branch that is consist by 312. So 312, 305 and 309 makes a cluster comparing to 322 or 295 or something 330 or whoever present in the whole tree.
Sir, this 307 and 306 are more closer when compared to 305 and 309, right?
So shall I try one more time?
No, sir, 1 minute, sir. Let me ask my question. This after
Okay.
this we can. 307
Now
and
you
30.
tell me, I ask the question. Maybe you can tell me, I can ask the question.
Okay, sir. Okay.
So I'm asking whether 329 is more closer to 330 or 334.
330.
It is more closer to 334.
Yes, sir.
Okay, so whether 327 is more closer to 295 or 306.
327 is more. I'm
More
not
closer to 309 here or 306.
309. I couldn't find where it is.
309 is here.
Yeah. Sir, actually, this cluster is closer one, so this is far away because it is bypassing here. Related members, and this is not a related one.
So you can try to make this tree in several ways. You know,
Yes, sir. Yes, sir. Yes, sir.
like you can go to the view.
Yes, sir. I know.
And then click on tree branch style. Use the radiation, use the circle. I mean, result will be same,
Yes, sir.
but just to explain the tree will be different. The way of explaining the tree is different.
Sir, what we can talk about means which one is the origin or the first ancestor in
No,
terms of this?
you cannot place here the root. This is unrooted tree.
Yes, sir.
To place the root, you need to find someone with a designated mark that this one is the ancestral to them. For example, like if you are comparing whole human population, then we know that the Africans are the one where we all collies. I mean, Europeans or Indians or Chinese, everyone goes to Africa. So we can take Africa as a common ancestral route. So
So
from
that should
Africa,
be a known one, biologically identified as ancestor. Only we can mark that as root.
exactly. I mean, placing a hypothetical route usually doesn't work. Most of the time, the real route usually works.
Okay. So if I place things. Okay. Can I stop sharing it now?
Did you tell something?
Can I stop sharing it now?
Yes, please. Yeah, please. So does anybody else have the problems in making the tree? Then you can share the screen and we can rectify. Or
Yes,
if
sir. I
everyone
had.
and if everyone is happy, then I'm also
I'm
happy.
unable to prepare the tree because it's.
You cannot share?
Sir, am I audible to you?
Yes, yes.
Yes, I already shared, but I don't know.
Your tree is not visible. We don't see any sharing. Oh yeah, now it is coming up. Still, we cannot see your. Only your picture is coming. Can you please try again? Oh yes, looks like now we can see your. Okay. Now go to phylogeny. Yeah, phylogeny. Click on never joining. Yes. Yes. Click yes. Click on OK. You don't need to change anything. Distance matrix could not be computed if you selected the Oh yes. You made a mistake. You can go back to your alignment. Can you go back to your alignment? Click on align. Click on align. Click on edit build alignment. I can make a new one. No need to. Where is your alignment session? Click on all files. Yes, SCQ. Okay. Open. You can make a new one. Edit build alignment. Yes. Create a new alignment. Create. Yeah. Okay. Yes. DNA. Yeah. You need to first click on the sequence. Click on the sequence 1 on the left side. Right click and delete. Yes. Delete. No, it has not been yet. Yeah. No, nothing. Yes. Now go to edit. Go to edit, please. insert sequence from file. Yeah. Yes. Alignment, then align by cluster W. Click on OK. Okay, export, mega format. You may give a name or whatever. New alignment, yeah, maybe. Whatever you say. Yeah, new file is fine. Click save. Save, okay. No. Okay, good. Now close everything. Close everything. And now click on the new file. Double click. Okay, now go to the. Yes, close everything. Close. Would you like to create or no? No, no. All mega is closed. Now double click. Okay. Are you sure? Why it is asking? Are you sure? Yes. Click OK. No, distance calculation failed, could not be computed. The no common site found for sequence 39. Looks like there is some problem with your original file. Did you open the file and made some changes? Maybe you can re-download that file.
No, sir. I didn't make any changes. The file is the same.
So it shows that this 39 number of
Okay,
sequence
sir.
has problem. You can look this tutorial, try again, and it doesn't work, then you can just directly call me, because there is no need to waste others' time.
Yeah, thank you. Thank you so much, Gyan. Ziol, I understand, you're able to, you're trying to do this. I would appreciate, you do the exercise, and if at all, if you're not able to do it, please paste a screenshot in the group so that, we can get back to you. Okay? Yeah. We cannot, work individually, looking at each and everyone's screen. Gyan has already been there for over 2.5 hours. Okay? We should appreciate his time as well. Yeah. Thank you so much, Gyan. I mean, without any hesitation, I think we would like to, thank you, for the wonderful talk, that you have delivered. It's been a learning experience, for many. Friends, instead of virtual claps, I wish everyone, to unmute your microphone, switch on your camera,